We are starting the fourth session of Fellows Talks of this annual Fellows meeting. We'll start off with first presentation by Dr. Ajaz Wani. He's our intermediate fellow at University of Kashmir, and he'll be talking to us about unraveling the principles of genome organization.
Okay, so it's a great pleasure to be here with all of you today, and thanks India Alliance for this opportunity. So we are working on understanding the principles which govern the organization of genome within the nuclear space. Okay, so the problem which we are addressing is how the DNA of about 2 meters long is packaged within the tiny cell nucleus of about few microns in size. So this problem is not only interesting from the perspective of dimensionality, but it's also interesting because it has the organization of the genome is linked with very important biological processes like cellular differentiation. So as we know that our development starts from a single cell, and as we grow, we develop different kinds of tissues. So even though these cells, they have the same DNA, but they have a very distinct identity. So what we know now that the packaging, even though these cells have the same DNA, but the packaging of the DNA varies across these different cell types. And if there is a perturbation in the organization of the genome, it can result in developmental defects. So the point here is that proper 3D organization of the genome is important for proper growth and development of an organism. So currently, the kind of an averaged out picture what we have about the organization of the genome is that it's a very hierarchical, the genome is packaged in a very hierarchical manner within the nucleus. And it's a multi-scale organization, scaling from the nucleosomes up to the chromosome territories. So nucleosomes are, in each nucleosome, about 147 base pairs of DNA are wrapped around the core of histone molecules. But this level of packaging is not enough to accommodate the DNA, all the DNA within this nucleus. So it needs to be packaged more and more into the higher order structures like in the form of loops, which further get packaged in the form of these sub-topologically associating domains, and then topologically associating domains, and several of these topologically associating domains come together and form compartments which can be active or inactive in terms of gene expression. And finally, each chromosome occupies a very specific place within the nuclear space, which we call as chromosome territory. Okay, so and these domains, which Wendy introduced in the morning, so they have become sort of units of higher order organization. Okay, and DNA elements within each of this domain, they have a high contact frequency among each other within this domain as compared to the DNA elements in a neighboring domain. And the two consecutive domains are separated by a region which has very low contact frequency called the boundary elements. And in my lab, we are excavating, we are interested in understanding these different levels of a genome organization. So Fozia and Yunus, who are the two PhD students, they are interested in studying the chromosome level organization. And Suhail is interested in understanding how these subdomains and these stats are stabilized or formed, while Zafar and Alfar, they are interested in studying more higher order organization. So as you can see, this packaging and organization of the genome involves the formation of a lot of these folded modules. Okay, so the question, one of the questions which we ask in the lab is what drives or stabilizes these different folded modules of the genome. Okay, and we try and we ask it from a very, starting with a very simplistic assumption that if the two elements of the genome have to come together to form a loop, okay, there has to be something which is physically holding the two non-contiguous regions of the genome together in order to give rise to the formation of this very simple structure or a loop. Okay, so and this can be done by the proteins which are decorating the genome, hundreds and thousands of them. Okay, and say these two red dots, they represent two proteins which can be sitting on two different regions of the genome, and they can come together by protein-protein interactions which are very specific and give rise to the formation of this very simple loop, which is involved in the, which is, I mean, these are several these formed within the packaging of the genome. Now, if we extrapolate this simple concept on a genome-wide scale, where there are hundreds and thousands of these loops and contacts between different non-contiguous regions of the genome, so it suggests that there has to be an extensive protein-protein interaction network giving rise to the 3D, shaping the organization of the genome within the nucleus. So this kind of a network, on one hand, it can give rise to and can act as a mechanical scaffold, shaping the genome architecture. And on the other hand, it can act as an interface between the external signals and the response from the cell in terms of an altered gene expression. For example, if a signal from outside induces a post-translational modification on any of these proteins, which can in turn perturb the protein-protein interaction, leading to a change in the underlying genomic contacts, and hence it might be associated with the change in the gene expression. Okay, and in the rest of my presentation, what I'm going to demonstrate you is that how disruption of a single protein-protein interaction changes the underlying contacts, genomic contacts, and what are its physiological consequences. Okay, so we have excavated a very tiny protein-protein interaction network, which is associated with the genome of Drosophila. And this network, as you can see, it has several of these domains. Like one of the domains is formed by the polycomb group of proteins, another one by nuclear pore proteins and chromosome condensation proteins, and the transcription factors and insulator proteins which bind at the boundaries of these topologically associating domains. So, and what we are trying to understand is how the physical links between these proteins help in shaping the genome architecture. So, as you can see here, in this corner, there is a one small protein-protein interaction, which is mediated by this pH protein. So, as you can see, this pH protein, it can self-associate. Okay, as you can see here, there is a curved line over here representing the self-association of this protein. Okay, so and this self-association of the protein can result in protein-protein interactions between this complex of polycomb proteins, okay, in which it is a part. And we asked, what if we perturb this protein-protein interaction, what will be the effect on the organization of the genome? And according to our hypothesis, the protein has to first be bound on the genome, and secondly, the protein molecules have to interact among themselves in order to drive the formation of these different folded modules of the genome. And we used a method which is based on the next-gen genome-wide sequencing to identify whether this protein is bound along the genome, and we found there are about 4,000 sites on the Drosophila genome to which this protein is bound. And to find whether this protein has a role, as our hypothesis is suggesting, in shaping the architecture of the genome, we used a method called chromosome conformation capture, again explained by Wani in the morning. And in a variant of this method, you can select a bait sequence and ask where else it interacts with the genome. Okay? So we did this experiment in a normal situation where this protein-protein interaction is functional, and for the sake of simplicity, you can consider the presence of red bars as the presence of contacts between the bait region and the rest of the DNA elements in this region, and the blue bars as the absence of contacts. as you can see, there are several of these red bars in this region, implying that the bait region is making several contacts between different regions of the genome within this fragment of chromosome 3R. And we did the same experiment in a perturbed situation where we disrupted this single protein-protein interaction by making a very specific point mutation. So what you can see here, the contacts with the more distal region are gone, and the sort of contacts within this region are enriched. So what appears from these data is that in case of a normal situation where the protein-protein interaction is functional, this entire region is folded as a single module. But when we perturb this protein-protein interaction by making a very specific point mutation on this protein, okay, so this sort of region, it splits up into two. And this was reflected also in terms of the subnuclear distribution of these proteins. So as you can see, in a wild-type situation, we observed these nanoscale clusters varying in size from 30 to 600 nanometers. But in case of the mutant situation, these clusters were perturbed, dispersed. So from this data, what we can take is that in a normal situation, in a wild-type situation where the protein-protein interaction is functional, you have the formation of these subnuclear clusters, which in turn drive the formation, which in turn drive the contacts between the different regions of the genome on which they are sitting. And if we perturb this interaction, disrupt this interaction, there's a disruption of this formation of these subnuclear clusters, which in turn leads to the loss of contacts between different genomic regions. So far, I told you how this disruption of this specific protein-protein interaction perturbs or alters the organization of the genome within these scales varying from subtads to the compartment level. We also wanted to check whether this perturbation or this disruption of the protein-protein interaction has any effect on the organization of the genome at the nucleosome level. Okay. For this, we used a method which is based on the micrococcal nuclease digestion coupled to the next gen sequencing. So the principle behind this method is that if a nucleosome is an environment which is less accessible, then there will be less digestion of this genome. But if the nucleosome is present in an environment which is more accessible, then this enzyme can digest more. Okay? So we did this experiment in, sorry. So, okay, so we did this experiment in both okay. Okay, here we go. Yes. So we did this experiment in both wild type as well as the mutant situation. And instead of using one concentration of MNase, we used a titration of MNase to make sure that we digest the most open chromatin as well as the chromatin which is highly compact or inaccessible. And one more reason to this is we can derive a parameter from the by using a titration of MNase, which we call as MNase accessibility score, by plotting the reads of the DNA sequences corresponding to each nucleosome against the MNase concentration, and you fit it to the straight line, and the slope of this line will give you the MNase accessibility score. And this is the plot, which shows you that there are a lot of differences between the wild type and the mutant situation in terms of the accessibility of these nucleosomes. So suggesting that this mutation which perturbs this very specific protein-protein interaction it doesn't affect only one level of genome organization, but it perturbs the organization of genome at multiple scales. So this change in the genome organization was associated with the change in the gene expression, and I'm not showing you the data for that. And this mutation, which I showed you, affects the organization of the genome at multiple genomic scales. The same mutation was shown by other groups, has very important physiological consequences. So in case of Drosophila, it was shown that the same mutation leads to an overgrowth in one of the spatial organs of Drosophila, and exactly the same mutation leads to the abnormal development of the skeletal system. As you can see, there is a presence of an ectopic rib on the C7 here in the mouse, which has exactly the same mutation, which we showed that this mutation perturbs a very specific protein-protein interaction and changes the underlying genomic contacts and is associated with the change in gene expression. Okay? So what you can take home is that we are investigating, we are trying to understand this mechanical framework of genome organization and the subnuclear distribution of these proteins and how these two are related in shaping the architecture of the genome at multiple scales. And we are also monitoring, whenever we perturb the organization of the genome, we monitor what are the changes in the global gene expression by doing RNA-seq. And then we try to see whether we can make an organization functional relationship and ultimately what might be the physiological consequences of this. So, more aspects what we are exploring is what is the role of these different heterotypic protein-protein interactions in this network which we have in shaping the genome architecture by using, again, biochemical next-generation sequencing and polymer modeling-based approach, which is on the more, progress we have made on this is on the poster. Please feel free to drop in. And what Fozia is trying to do is, given the hierarchical nature of this organization, what we are thinking is if we perturb a lower level of organization, do we see change at the higher order organization? So what she is trying to do is to change the spacing between the nucleosomes, which in turn will change sort of a persistence length of this fiber, and then try to find out whether we see change at the higher order organization of the genome. And what Yunus is doing is has reconstituted an in vitro remodeling system consisting of this remodeler I switch and the nucleosome, and he is trying to understand the mechanics of this nucleosome sliding by using isotope exchange coupled with mass spectrometry. Okay, and acknowledgements, I already told you who is doing what in the lab, and my collaborators, David from University of Edinburgh, who is helping us with the polymer simulations, and Jacob, who is helping us with the analysis of the next-gen sequencing data. And of course, the backbone to all of this is the award of intermediate fellowship from the India Alliance. Okay, as almost every experiment in the lab ends up in a next-gen sequencer, and which costs a lot of bucks. And without such an award, I won't even dream of doing these kind of experiments. And thank you all.
We have time for one question. Yeah.
Nice talk. So the protein-protein interactions that you are looking at, are you also correlating them with the epigenetic signatures that you have seen? Because, for example, the pH proteins, those are mostly with silenced.
I am not able to hear.
So are you also correlating these protein-protein interactions with the epigenetic signatures that are there, and when you perturb them, those also change?
So do you mean modifications, histone modifications? Yeah, I mean, basically, I am not interested in that aspect right now here. But one of the proteins, like polycom group protein complexes, some of them do have, enzymatic activity, and one of them has K27 trimethyl transferase activity. Okay, so and I'm trying to avoid those proteins which have the enzymatic activity, because it will be very difficult for me to dissect whether the effect in the organization of the genome is because of the enzymatic activity or whether it is because of the physical protein-protein interaction. So that will be very difficult to dissect for me if I work on those kind of proteins. So I am working on the proteins which do not have the enzymatic activity, which are not involved in the post-functional modifications of histones, so that I can say with confidence, okay, it's purely because of the protein-protein interaction.